So while we're waiting, I just want to introduce myself. My name is Angel Sampson. I'm the director of the Monoclonal Antibody Core here at the ICBR on the first floor of the Cancer and Genetics Research Complex. And we've invited Isoplexis to come and talk about their functional proteomic profiling platform. We do not currently have this instrument, but this is an instrument that we're looking at. And so if you have any interest, please send me an email after this seminar, and we'll see what we can do about coming up with a plan. Thank you.
Thank you, Angel. So I am, and we'll get started now after that kind introduction. I'm Jim Rainey from Isoplexis, and I am the Senior Sales Manager here in Florida for Isoplexis. So I'm responsible for all the sales activities that go on in the state. And I will be giving the presentation today, talking a little bit about Isoplexis and our platform and how we might be used in your research. I'm joined today by my field application scientist, Rose Lawson. Rose, you want to introduce yourself?
Oh, hi, my name is Rose. I'm the field application scientist for the Southeast. I'm really excited to talk with you all. I'm going to be giving a data presentation, and I'm looking really forward to it.
All right, so we'll see now if I can be technically adept enough to share my screen without a hitch. I'm going to take my ugly face off of the screen and stop inflicting that on you, and instead give you a lovely presentation to look at. So. All right, you guys should be seeing my screen right now. Are you seeing the presentation?
We see it.
Excellent. Okay, so thank you everyone for being here today to share your time with us this afternoon. Today we're going to be talking about Isoplexis and powerful biology accelerating the future of medicine using the Isoplexis platforms, the IsoLight and the Isospark. We've just done quick introductions. So you've seen me, I'm Jim Rainey, and Rose, you just met. And so we'll have about a half hour of the presentation per slide deck, and then Rose will go through the IsoSpeak software, which is an integral part of the system, and then we'll have some time for questions and answers at the end. Before I get to talking and filling everybody's heads, some of you may have, because you've seen the flyer and we're prepared for this, might have taken a look at some information and have some questions already. So I'm going to stop real quick and just ask if you have any particular questions on your mind or any information you were looking for specifically, just go ahead and shout it out and let me know what you think at this point. You can also type your questions into the chat function or the Q&A function on the system, and we'll see them. So feel free to type questions at any time during the presentation into the chat or into the Q&A, and we'll try to handle those as they come. Otherwise, again, there will be a question and answer period at the end of the presentation. So I'm going to start with a couple of quotes that have come from recently published studies that were using our technology as part of the data gathering for the studies that they did. Both were in Nature Medicine. One was published in July of 21, and the other was published in August of 21. So as you can see, we have very recent publications covering our technology. In fact, it's becoming an almost weekly event where we're seeing a new publication in a well-respected journal using our technology. So the first quote was from a study that's Spiegel that was done. These folks were collaboration with Stanford and Loyola University School of Medicine in Chicago. And the quote from them is, "Our results suggest that the engineering iterations should be guided by careful studies of single-cell CAR polyfunctionality, incorporating cytokine production as a critical quality attribute." And then this is an attitude we're seeing more and more folks who are working with CAR T cells. The second quote is from a study that was done out of Moffitt here in Florida. This was a collaboration between Moffitt and Duke, and it actually had a sort of a tie-in with the University of Florida, because Dr. Kay over at Shands Cancer Center over there was involved with this study. And in this one, they said, we assessed the polyfunctional strength index, PSI, which is an indicator of the number of cells capable of secreting multiple types of cytokines. As PSI reflects the ability of a T cell to carry out multiple functions, it is recognized as a metric for the potency of cell therapy and for the efficacy of vaccines. And indeed, when you take a look at the literature, and if you Google Isoplexis, you'll see quite a bit of peer review literature that you can review. PSI is a metric that you'll find coming up again and again because it's unique and predictive and provides powerful information in those studies. Because we're providing such valuable data from our platform, we're growing very rapidly. So Isoplexis is a younger company. We just actually launched our initial public offering just a couple days ago. And we are getting, we started placing instruments in 2018. We had been operating as a services company before that, doing the testing in-house and giving customers the data so that they could work with it. But in 2018, we started placing instruments. And since that time, we have over 155 systems in the field. There are actually north of 90 now current publications in the peer review literature. And we have over 15 total products and 12 distributed partnerships across the globe. So we're growing rapidly and we're being rapidly adopted by many high profile researchers. And you can see that our unique data is accelerating cell therapy for a lot of folks, a lot of well-known and well-respected institutions on the slide here. And we're being used in the preclinical space, in bioprocessing and clinical products. You'll see in preclinical, we're working with places like Nectar, with Moffitt, where we have a platform, with NIH, with Baylor and UCLA to do the predictive testing before products go into trials. We're working in the bioprocessing space with Lonza, who uses us as a quality check in every step of their cell line productions. Also with Iovance here in Florida, one of those published papers, the one from Moffitt also included a collaboration with Iovance. And then for clinical products, Kite Pharma was an early adopter for us and used us in their studies. The NIH, Duke, and Moffitt all are using us. And in fact, in the clinical product sense, Isoplexis's data was used in the data packages for the first two CAR T products that were FDA cleared. We're predictive in CAR T, in NK cells, in macrophages, and TCR-T, and it's across allogeneic, autologous, bispecific, and CRISPR modalities. The system itself, this is a picture of our Isospark, which is what we think of as sort of our academically sized instrument. That's about 18 inches on the side, so it can fit in most researchers' labs, even where you're crowded. And we think of it as the Swiss Army knife of functional proteomics. The Isospark and the IsoLight, its larger sibling, all do three unique applications. So they have multiple functionality for a single platform. They can perform low volume, bulk, highly multiplex testing. So this is your bulk cytokine analysis like you might be doing today on something like a Luminex. And the unique thing about our product is that it's very low volume. It only takes 11 microliters to do a pair of technical replicates. And it's completely automated so that once you put the sample on the experimental chip, you just put the chip onto the system and walk away. You come back later and all the steps that are done in an EIA test, all the washing, addition of capture antibody, washing, addition of signal generator, all that's done automatically. And that system then automatically sends all the data across to the IsoSpeak software that Rose is going to demonstrate later. So same day, in a couple hours, you're going to have all of your data crunched and publishing ready graphics. The second application that it has is the predictive immune single cell secretome. That's that functional immune phenotyping that we do. In this case, we're going to take cells, we're going to incubate them on the experimental chip for about 13 hours and let them excrete the cytokines that they would produce in vivo, and we measure those cytokines. All three of these applications are highly multiplex, so you're going to get as many as 32 different cytokines measured from a single sample. And finally, the last application is predictive tumor pathway mapping, where we'll look at the single cell phosphoproteomics. We'll actually look at what are the active phosphoproteins inside those tumor cells so that you can understand the tumor signaling pathways. By combining all of these different applications, we can help accelerate and advance the medical industry. So how does it work? Well, this is a little bit of a simplified analogy, but it works to help people understand. So if you think about if you were to make a fruit smoothie or other homogeneous sort of beverage, all the ingredients that are going in this case, individual fruits, they're all ground up together and you're going to get data across averages. So if we're doing bulk cytokine testing, like you might do on a Luminex, you're going to have a sample that's a bulk sample. You're going to get the cytokines that are produced and you'll see the spikes. And perhaps you see a spike at granzyme B, but that's for the whole sample. We don't know what immune cells are making that and how much. And you might find when you look at the single cell level that you've got a lot of cells making just a tiny bit of granzyme B that's making a spike, whereas you might have an IL-2 spike on some individual cells. And those are the things that are actively affecting the process that you're studying. So Isoplexis functional difference is that functional phenotype where we capture the cytokines from each individual cell, again, up to 32 different cytokines for each individual cell that we're studying. And we can successfully capture what we like to call superhero cells, those cells that are predictive of response. These are the cells that are polyfunctional, producing 2 or more cytokines. And these are the cells that are effective in the pathologies that we study. If you're just looking at some other technologies like bulk ELISA or flow cytometry, which are excellent technologies, they can't see that functional phenotyping. And so they may miss critical pieces of information that are predictive of what a clinical candidate will do or predictive of a response of patients. So when we're looking at that functional phenotype to reveal those superhero cells, we're uniquely and highly multiplexed for over 30 cytokines from each cell, which is going to reveal different types of cytokines that are being produced. We'll be able to tell whether it's effectors or chemoattractors or inflammatory, et cetera. And we'll be able to tell if that cell is making multiple cytokines. And from that data, we produce a parameter called PSI, which we referenced in the earlier quote. PSI is a parameter that's simply taking the percentage of cells in a sample that are polyfunctional, meaning they make 2 or more cytokines, multiplying that times the strength of that signal of that cytokine production, and that gives you the polyfunctional strength index. You'll see that parameter over and over in the peer-reviewed literature, and it is predictive. It's a unique value that only we're producing, and it's predictive. You can see in this graphic, it's showing you responders and non-responders, and in a moment, we'll get to a study where this data actually came from, and you can see what was happening there. So before I move on to the studies, I don't see any questions in the chat, but I'm going to stop for a moment and see, do you have any questions at this point? Please go ahead and type them into the chat for the Q&A, and we'll answer them as best we can. Okay, I'm not seeing any questions, so I'm going to plow on ahead. So let's look at a study. As I said, we're going to show you where that graphic came from. Again, in this graphic, you're seeing the polyfunctional strength index on the y-axis, and then you're seeing responders and non-responders on the x, and you can see there's a significant difference. In this study, it was looking at CAR T products. This is from a Kite Pharma study at NCI, and they were looking at, if we're looking at that functional phenotyping, what are we going to see that we might have missed with flow and some other technologies that we're using? This is from a 20-patient study looking at non-Hodgkin's lymphoma, looking at CAR T products for it. And they wanted to see before infusion of the CAR T products, what the response would be. And the polyfunctional strength index was able to predict the responders and non-responders before infusion. And if you look at the other parameters, the parameters that in this case were being produced by flow, this is p-value now on the y-axis. You can see that PSI was the only parameter that had strong p-values in this study. This is another study, and this is the one that was out of Stanford, and they were looking at what are we missing by ignoring cytokine signatures, looking at critical cell pre-infusions. They wanted to know if we look at our cells products pre-infusion, can we predict whether they'll be effective or not? In this case, we're looking at stimulation for CD19 or CD22 in CD4 cells and CD8 cells. And as you can see, there's a significant difference between the responders and non-responders in the PSI in terms of their strength of response to the stimulation. And the results suggested that engineering iterations should be guided by careful studies of single-cell CAR polyfunctionality, incorporating cytokine production as a critical quality attribute. This is a study that came from leaders at NCI. They were looking at, can we be predictive? This was a mouse study in hepatocellular carcinoma, and they wanted to see if they could get data that was predictive before the trial started. And so it was in a solid liver cancer, again, working in a mouse model. And you can see the top bar, so it's a heat map looking at healthy donors. And then the bottom is the patients. And then you see the mock T cells, HN3 and HYP7, and the response. The orange bars are just the darker the orange, the higher percentage of cells that were polyfunctional in that subset. And then the blue bars down here, you're seeing the individual sets of cytokines, what combination of individual cytokines is being produced. And what you're seeing here in the HYP7 is what we like to call a long cytokine tail. I mean, you're seeing a lot of polyfunctionality, a lot of cytokines produced, and these tend to be predictive of responsive cells. And you see the same kind of data in the patient samples. Again, with the HYP7 sampling, you're seeing a long cytokine tail that was predictive of response. And indeed, that's what they saw. This went on to a 10-week study in mouse samples, and in the mouse cohort that had HYP7 that was infused with 20 million cells. The entire cohort of the mice was survived out to the 10-week study endpoint, and that was the only combination that did so, and it was completely predicted here. And so they knew 10 weeks before the mouse data was back exactly what was going to happen with those CAR T products. And that's why they got to the point where they were saying the polyfunctional persistence or persistence of polyfunctional chimeric antigen receptor T cells that target the glypican-3 eliminate orthopedic hepatocellular carcinomas in mice. It provided them a framework of targeting, and it accelerated their research. So they were going much faster. 10 weeks before the end, they already knew where they were going. This is a study that came out of Moffitt, which I'm very fond of because it's where we have a platform today already here in Florida. In this case, we're looking at what could they learn from the T cells by doing that functional phenotyping that they might miss with surface phenotyping or other methods. This was a lung cancer study, small cell cancer. It was an Evo combination. So in this case, they were testing PSI at baseline. Then they would test it post-Evo administration. And for the patients who did not respond to Evo, who still had tumor growth, they would move on and do TIL infusions. And then they tested PSI both at the 0 time point at TIL infusion and then 4 and 12 weeks post-infusion. And what you can see here is there's a significant bump up in polyfunctional strength index after the TIL infusion, and it's a durable response out to 12 weeks. And again, they saw a good response in the patients in the study. That's why they said what we quoted earlier today, which is as PSI reflects the ability of a T cell to carry out multiple functions, it is recognized as a metric for the potency of cell therapy and for the efficacy of vaccines. So how does this kind of all work together? Actually, let me stop again for a moment and see if there are any questions based on the studies we just looked at. Any questions, comments, feel free to put them in the chat or in the Q&A function. All right, so not seeing any questions at this point. So again, I'll keep moving forward. So when we do this testing, we're looking at a simple workflow to a sample answer on one system. And again, it's from the time the test chips go on the platform, it's walk away, hands off. So we can use many different cell types. They need to be cryopreserved or fresh. And the reason for that is for the single cell secretome, for that functional immune landscape phenotyping that I'm talking about, we need to get viable cells. So what we do is we take those cells, and there's some steps up front. You're going to do an enrichment, you're going to do some sorting, you're going to do a staining, and then you're going to put them onto these experimental chips. The experimental chips then go onto the system. Either the IsoLight can run 8 of them at a time in this slot at the top of it. The Isospark could run 4 of them at a time. And then the system does all the work from that point. It does all the sample additions. The reagents for the whole test go into the bay at the bottom here or on the side here on the Isospark. So once you load your reagents in your chips, you walk away, you come back the next day and you get your results. The results, when they're done, are going to go across to our IsoSpeak system. And that's going to do the data analysis, the kind of data analysis that today you probably send off to a biostatistician, or maybe you do the crunching yourself and it takes weeks or months. I had one researcher, it was taking him a year to get his data back. And instead you're going to get it back in a couple of hours. And you're going to get everything from the raw IFUs all the way out to publishing the graphics with things like 3D Tisneys and bar graphs and everything you could want. The way the system works within the chips is we're going to try to load about 30,000 viable cells onto the chip. On each chip for the single cell secretome products, there are 12,000 microchambers. Each of those microchambers is going to try to capture an immune cell. So the cells are going to distribute in a random Poisson distribution, meaning that some of the chambers are not going to capture a cell. Those are used for a background read. Some chambers will capture multiple cells. The system recognizes that and ignores them. The system will only look at those individual cells in an individual chamber. Those cells are incubated, well, all of them really are incubated for 13 hours on the system in near physiologic conditions, and they excrete the cytokines that they would naturally excrete in vivo. Those cytokines are captured on a microarray, what we call a proteomic barcode. It's up to 32 different capture antibodies in the microarray, and we capture the antibodies, and then the signal is generated, and then that data goes across to the Isoplexis platform. It's highly parallelized cellular proteomic reactions. It's flexible with 13 current panels and 19 panels in development, delivering actionable functional biology from the most powerful subsets of cells. And this is kind of a look at the Isospec system. I'm not going to go into detail here because Rose is going to take that on and show you all the features on that system. So we're just going to move on past that. So when you think about, again, remember, this is a multifunctional platform. So in a week of testing, you can deliver predictive data, from the Swiss Army knife, meaning you can start on day one with that highly multiplexed bulk cytokine analysis and get that running on the platform while you're preparing your cells for your single cell secretome experiment. On day 3, you can get the functional immune landscaping from the single cell secretome, getting that PSI, that polyfunctional strength index data. And then finally on day 4, you can run the intracellular signaling omics, and you can get the heterogeneity data showing you the signaling pathways in the tumor that you're looking at, and make some decisions based on that. All of that powerful, unique predictive data from one platform in just a week of testing. So here's some more studies showing what can be done with the system. This is again working with the single cell secretome. In this study, they were looking at metastatic melanoma. This was with Bristol Myers Squibb and Nectar Therapeutics, and they were using a combination checkpoint inhibitor and IL-2 agonist therapy. And this is interesting data because it's predictive of survival in these patients. What they did is test the patients at day 0 and then again at day 8. So they had a polyfunctional strength index on both those timelines, and they looked at what they called a polyfunctional strength difference, meaning what's the difference in strength between their testing on day 1 and day 8. And what you see is on the red line on the top, so it's the probability of progression-free survival on the y-axis and months on the x-axis. The red line is where you had a CD8 polyfunctional strength difference that was high, 0.71 or more. The blue line is the CD8 polyfunctional strength difference that was low, 0.32 or less. And you can see the survival out to timeline. On the high PSD line, you're out to 36 weeks with almost 75% of the cohort having progression-free survival. Whereas you can see in the cohort that had the low PSD, it was dropping off very badly out to 32 weeks to an endpoint there. So highly predictive of progression-free survival. Again, not only highly predictive, but highly predictive early at 0 and 8 days. This next study is a study that's done in COVID. In this case, they wanted to know, could we predict the severity of the case, of the disease course in our patients? And so they tested with, again, the polyfunctional strength index, and they were looking specifically at the monocytes and the PSI in monocytes. And you can see for healthy patients, mild patients, moderate course, and a severe course, there was a significant difference in the polyfunctional strength index and in the cytokines that were being produced amongst these severe and moderate cases. So it was predictive, of course, for those COVID patients. We're seeing a lot of COVID studies being done right now. We just had another one come out that was looking at neurological deficit in those long haul COVID patients, and looking at the cytokine production using our CodePlex product, where you could define what was driving that deficit and the cytokines that were doing that. There's another one that we were looking at that was comparing the cytokines that were produced from the sample in bronchial lavage versus a peripheral blood sample. And it was stark the difference in the cytokines that were being produced in the two different sample types. So a lot of COVID studies are going on right now on our platform. And finally, this one is using our intracellular phosphoproteomic panel. And in this case, this was a study, ISB, looking at glioblastoma patients. And the challenge for them is they know from the genomic testing that you can identify the different signaling pathways that are causing resistance. There's a core set of mutations that are driving those resistant pathways in the tumors. However, when we do design drugs or therapies to treat those core mutation pathways, you get resistance. And so we're looking at, can we look at the intracellular phosphoproteomics and map out those resistant pathways and then design treatment to attack those pathways? Indeed, that's what they did in this study. And this one, we're looking at with a glioblastoma model known to be resistant to TOR kinase inhibitors. And so they were looking at, in the graphic here, for different drugs. So we're looking at drugs that they found with the testing, looking at the heterogeneity index, which is like the polyfunctional strength index, only looking at phosphoproteins, for those that are predicted to be responsive or resistant. And so for all of these different drugs or drug combinations, they were able to predict which would be resistant and which would be effective. They predicted that just these three combinations here would be effective and the rest would not be. And again, with testing, this is tumor volume in the mouse over time on days. They started the treatments, and you can see for all the drugs that were predicted to be ineffective, that resistance would develop. Indeed, resistance developed rapidly, and you saw tumor volume growth. However, the three drugs that were predicted to be tumor suppressive were indeed tumor suppressive up to 47 days, at which point they discontinued the therapy and you immediately saw tumor growth. So it allowed them to predict early on, anticipating drug resistance long before it was clinically manifest, and combining therapies that were able to be effective at tumor suppression. And that is where I'm going to stop. Any questions about the technology or anything that I talked about in my presentation before Rose goes on to start talking about the software, which people get just as excited about the software as they do about the technology. So I'm not seeing any questions or any comments. So Rose, I'm going to turn it over to you to take it away.
All righty, I will go ahead and share my screen. Give me just a moment. All right, can everyone see?
Yes.
Perfect. All right, so this is the IsoSpeak software. This is the first page. So this is what you would see after you've loaded in your experiments. So the first thing I want to highlight here is this is a CAR-T demo data set. So this is real data that has been de-identified for purposes like this. And if you'll remember in the presentation, Jim mentioned that the machines have different throughputs, so an 8-chip or 4-chip throughput, but that doesn't mean you're limited to that throughput in your data analysis. So you can combine up to, I believe, 500 chips in one data set. That would be quite a hefty data set. This one is 24, so they were looking at, non-responders and responders. So yeah, that's pretty much the first page. So I'll jump us over to biomarker analysis, to some of these 3D TISNEs that Jim was talking about. So right now, this is sorted for stimulation condition. So we've got two different stimulations. There's the CD19 and the NGFR stimulations here. And you can actually toggle all of these proteins. So right now I have it selected to the secreted. So these are the true secreted proteins from cells, and that's in this sample. And something else that's really interesting that you can do is you can actually hover over or mouse over each individual cell. So each dot here is one cell that was detected in the assay. And I'm trying to find one where you can see some secretions in it. Let's see, here we go. So you can see here in the right-hand side, you can see where that cell came from, what the experiment ID is, what cell subset it is, the donor, and then the proteins that were secreted from that cell. So in this cell that we have highlighted here, this cell was secreting MIP1A, and that's something that you can do more with visualizations here. You can change and sort this by polyfunctionality, dominant functional group, or some of these other metrics. It can help you glean more from your data than just looking at dots on a screen. So another version of that, we've got a PAT PCA, and that's going to be a more linear view of the 3D TISNE. And I quite like this because it groups things really nicely. So they're grouped by the overall color of that sample. So each sample here has a different color in the key. And each dot here is one cell. So this is a more linear version of a 3D TISNE. And everything that's further away from the organ is going to be more polyfunctional or secreting more unique combinations of cytokines. So you can see these groupings here have some unique combinations of cytokines, and that can help you come up with further hypotheses for your work. We also have this polyfunctional heat map that Jim was showing some in the presentation. So this one's got quite a bit of information because it's got 24 samples on there, but just to give you a little bit of bearings. So here, starting on the right-hand side, left-hand side moving to the right, you're going to be going from less polyfunctional to more polyfunctional. But in fact, all of this is going to be pretty polyfunctional. So it's going to be 5% to 0.1% of the sample that are secreting these unique combinations of cytokines. So you can come down here to the key and see that this sample here was secreting both IL-9 and granzyme B, and that's a unique combination. And the combinations of these cytokines are what's driving that polyfunctional strength. So speaking of polyfunctional strength, we can jump over to that. So this is going to be that polyfunctional strength index. And before I kind of dive into this graph, I want to show you two others that are used to create this measure. So first we have the polyfunctionality. So this is going to be the percent of your sample that is secreting 2 or more proteins. And you can see the percentage here. We also have a lot of options in the software. You can log transform, you can normalize by viability or transduction, you can show fold difference, you can have the error bars, you can change the values here so that you can set it how you want. You can also change how the samples are labeled and sorted. So it really gives you the flexibility you need as a researcher for your research story. So this polyfunctionality, this is the percent of the sample that's secreting 2 or more proteins. And then multiplied by the signal intensity of that secretion, so how intense those secretions are, those two give us this PSI metric. So that just gives you a little bit more understanding to what goes into this. And this is that metric that is, kind of our claim to fame. This is the one that has been shown time and time again in studies to be more correlative to patient outcomes. And in fact, they did see that in this study, that donor 5 treated with the CD19 treatment, had a significantly higher PSI than some other donors. So that helps them see their responders versus their non-responders and things of that sort. And then I think I want to show you one more thing. So we've got this here. This is the PSI per protein, and I apologize, it's a bit small. It's a lot of chips at once. So you can see you can even break this down by sample by protein. So you can see in, donor 4, CD4, the NGFR treatment, there's not a whole lot of secretions going on that are contributing to that PSI. We've got some that are being detected, but are not significant. But you jump over to the donor 5 treated with the NGFR, and you can see that there are secretions occurring, and then you can actually see what is driving that polyfunctionality strength index. So you can see here, granzyme B is the highest secretion. But if we go down to another, in this sample, donor 5 treated with the CD19, MCP1 is going to be the highest secretion, or the one that's contributing the most to the PSI. So you can really break it down and see what's going on in your sample and what your research is telling you. That's pretty much it for the software. I will show you a couple more things real quick though. So this is going to be that data overview tab, and this is where you can actually pull values for those graphs that I was just showing you. So you can pull the values for the PSI per protein, for the PSI, polyfunctionality, strength intensity, and all of the others. And you can actually run statistical analyses on these. And all you have to do is press export, and it'll automatically be loaded into an Excel file for you, or you can choose a text file if that's what you prefer. You can also do this with the raw data. Now with the raw data, this is also going to include those cells that were not single cells. So if that's something that's important to you, you can have that data as well. And I'll say this to you as well, that the data you generate is yours. We do as a company like to help design experiments with researchers just because it's a new technology. And sometimes we will have you send in the data if it needs to be quality controlled, but you own your data outright. So don't worry about any of that. I think that is pretty much it. If there are any questions, feel free to chime in or type them in the chat.
All right, so I'm not seeing any questions in the chat so far. At this point, pardon me, Rose?
We have a hand raised. I believe Christopher has a question.
Chris, did you have a question?
No, I was just clapping.
Oh.
Thank you.
Thank you so much. Anyone else who'd like to clap, feel free. But at this point, we're into the Q&A section of the program. You can type your questions into the chat, or you can raise your hand and ask a question, or yeah, I'm pretty informal. Feel free to just shout it out if nobody else is talking. So we've given you a lot of data to look at. I hope you've seen how our platform has been used to drive forward the research to give unique insight into the pathology or into the preclinical candidates that folks have been working with, and to see how we accelerate research and help folks move the research along faster. And I'm hoping that you might be thinking to yourself that you could see that in use in your own work. So far we've got no one the question. I know you are very, very smart people, but I'm surprised that nobody wants to know anything.
Hi, this is Angel. I have a question for you.
Yes.
I was wondering, Rose, could you go back to your presentation real quick?
Sure.
Sure thing. What's up?
And you were showing all these different options when you're, I guess, after you've loaded your data.
Right.
Can you go over some of those? Like, when would you choose one versus the other? I mean, what's the strength of that? Can you just kind of get into?
Sure. So it's really going to be dependent on your research and what you're looking to see. But a lot of times people are really interested in this PSI metric because it's the one that has been shown to be correlated to patient outcomes. A lot of people use this in their, not just one area, but I see this a lot in the vaccine studies that people use this PSI to show the difference between their treated and untreated groups, or vaxxed and unvaxxed. And that can be a really powerful metric to use there because it really helps you see which cytokines are really driving that response to that vaccine or that treatment. Others that people really like to use, the 3D TISNI, this one can be really, really helpful whenever you have the graph kind of tailored to the story you're trying to tell. So if you're looking at, you want to see if chemoattractive cytokines are driving this response, maybe the dominant functional group would be a better way to sort for this so that you can see if you have a huge prevalence of, chemoattractive cytokines or inflammatory cytokines, even, if you're working in inflammation work. It also is great to sort this 3D TISNI by polyfunctionality, because that's going to kind of separate the graph. I unfortunately can't show you all of those right now because it's a 24-chip file. So if I select any of these different things right now, it'll have to regenerate the TISNI. And I don't want to take up too much of your time just waiting on a graph to generate. And then this polyfunctional heat map is another one I see a lot of researchers use because they're really interested in those unique combinations of cytokines that are driving that polyfunctional response in their work. And I know that it's a lot, and I know that this is a lot of new figures, a lot of new terminology. So I totally understand wanting to figure out how to use these in publications. So the answer is all of them, but with a caveat in there of, we want to make sure that it fits your story. But that's something too that if I'm helping and I'm involved in this research process, helping design your experiment and things like that, I can also help you figure out which graphs would be the best to put in a publication based on the data we see.
Mm-hmm. And that's a point I'd like to make, actually, Rose. You've segued into something important. So when you purchase a system from Isoplexis, you get the system, but we're not just going to drop it off in the core and shake a hand, take the PO, and run away. You do get ongoing support with Rose, actually, is the one covering at the moment, but we're growing like crazy. So I can't promise we'll always be Rose because we keep adding and making smaller territories. But you'll get a field application scientist who really is good for helping to design experiments. I know everyone that we place, one of the first things that happens is, we've put the system in and we've talked about the approved and validated protocols that we have for lots of different things. But about 5 minutes after somebody gets the system, they think of a different way to use it and they want to try something else. And so Rose or another field application scientist can help you design an experiment that we know will be functional. So if it's something odd, I've got one collaboration we're working on right now with a very well-known institution where they want to take breast tumor samples that they have cryopreserved. They want to disaggregate them, get single tumor cells, put them on the intracellular phosphoprotein panel, and do some comparisons between drug treated and non-drug treated, or a couple different drugs, and see how that looks in terms of the active phosphoproteins. We don't have a validated protocol for that because nobody's ever done it before, as far as we know. So we're working together with them to design that. And that's the kind of thing that Rose can do with you guys or another field application scientist, so that we can help you create experiments that are going to get you data that's going to be powerful. Another really important thing that Rose can do for you is when you come up with that great brand new idea nobody has ever done, she can go, yeah, no, that's not going to work. We can't do that. So, because as much as I'd like to, as a sales guy, say it can do everything, it does have limitations. So Rose can help you see where the guardrails are on that.
So on this graph, I'm having a hard time understanding how the heat map relates to the cytokines listed in the bottom. Can you show that?
Sure. So these squares are going to be colored based on the percent of the sample that is secreting this combination of cytokines.
Oh, combination. Okay, got it.
Right. So in this first row, this is just one cytokine that's being detected. It's just TNF alpha. But there are others that are combinations, and it's going to be that percentage of the sample based on this key here, which color is secreting that combination of cytokines. And as you move further to the right, it's going to be more unique combinations of those cytokines. Does that help explain that for you?
Yeah, it makes it a lot. I understand a little bit more. And so the software automatically sets that up based on the results from your run?
Exactly. Yep.
Okay, that's cool.
It's a very useful graphic that generally takes more than one explanation for most people. So
Yes.
It just is. It's very busy, but it's very powerful.
Yeah, it's one of those times where I'm like, oh man, I'm trying to explain this in 20 seconds, something that took me 2 months to understand. So I totally understand. And I'm happy to explain any of the graphics, 1,000 times if you need it. So just let me know if something doesn't make sense. I just want to make sure that you understand and that it's clear.
So can you go back to the PCA graph?
Yeah, of course.
Okay, that's not the one.
Yeah, this one.
This one, yes.
Takes a second to get there.
Oh,
It does.
okay.
It takes a second to load.
So can you explain this a little bit more in depth? Because I was having a hard time understanding what I'm looking at here.
Sure. So this is just a linear version of the 3D t-SNE. Each dot here is going to be one cell, and each cell is colored from the sample it came from. So a pink cell is donor 6, CD4, NGFR. But these groupings are cells that were secreting more similar cytokines to each other. But as you move further away from the origin, those cytokine combinations are unique. So these groups are unique, but similar to each other, if that makes sense. So all of these cells here are secreting TNF alpha, but they may be secreting something else with TNF alpha. Same with these up here with granzyme B, and these ones over here with granzyme B, but these ones are further away from the granzyme B on the right-hand side because they're secreting, things that are not TNF alpha. And this one up here is grouped with TNF alpha and granzyme B. These at the origin are going to be less polyfunctional, and they're going to be secreting less unique combinations of cytokines. Does that kind of help you understand? I can go a bit further if you need.
So the dots are each individual cell, and that's based on the. Okay, I think I have it now.
Great. Any more questions?
It's a very quiet audience. Well,
So does your
go ahead.
website have a list of the publications that can you show us a little bit about your website maybe, and show us how we can get more information?
Sure. And the answer is, of course, yes, the website does. Give me a moment here to get the website launched. I wasn't ready for that question.
Sorry.
While I'm doing that, by the way, I'll let you know that for the deck that we've gone through together, That one will be made available to you guys so that you can get that. And then additionally, if you have any questions, my email is in the presentation. And so we can, sorry, I'm having some technological challenges here. There we go. Okay, I'm going to share my screen with you to show you the website. All right, are you guys seeing the website up?
Yes.
Okay, great. So this is our website at Isoplexis. Just to finish the thought on the presentation, any questions that you have about the presentation, or if you'd like the studies behind the slides or things like that, just let me know. My email, james.rainey@isoplexis.com, is on the last slide of the presentation, so you can always go there. So this is the website. It's much like you would expect a company website to be. We've got a listing where you can go in and look at products. If you wanted to go search by specific research areas, you could go in and see by cancer, immunology, infectious disease, cellular, regenerative medicine, inflammation and neurology and oncology. If you want basic science information about how the system works and that sort of thing, it's here. My favorite place to go is the scientific resources tab. Under the scientific resources tab, publications that you're asking for, you can view all the publications that we highlight. And I don't think we have every population in the world on here. They're just coming out too fast. And there's too many, but we have many, many of them, particularly ones that have information that's very descriptive for us. You can view them all on a list, or you can look for a particular research area. So if you were interested in cellular and regenerative medicine, you can just go in there, and then you can look at the publications that are under there. So it comes down here, and they'll have some called out that you can just scroll through and see, what titles look interesting to you. And then you can click through to get the actual document behind them. You can also go under scientific resources to Isoplexis University, where it's going to walk you through. You can be an Isoplexis field application scientist after a while, just walking through the different educational courses that are on here. Also, there's a certification you can get through the website for being a certified Isoplexis user. Product literature, under product literature and protocols, this is one where it's great to look at the different protocols that we were discussing. So if you wanted to go through and look at the different application protocols for different types of samples that you might run and how you would do it. So for the different panels that we have, you can see product sheets and catalogs and that sort of thing, but you can also go in and look at the actual protocols for how to prepare the sample for the experimental chip of your choice. There's lots of webinars and videos just broken down into broad categories you can look at. And then I really encourage you to go to the single cell proteomics blog. This is where we'll do a summary of recently published things that came out. For instance, this current one that we've got at the top of the list is from Cancer Immunology. It's looking at the single cell functional proteomics, revealing insight into optimization of radiation and immunotherapy. Maybe not the first place you would think to go with our technology. And there's all sorts of blog posts that they're generally going to highlight recently published papers or sort of new insights that have been developed in how you can use the technology to drive your research. So that's all there as well. Go back here. There's always on the front page the usual kind of marketing things you would like. Something very exciting here that I don't talk about a lot yet because it's not quite here, but this is our Duomics, where we're going to take on the experimental chips right now, where you can do the single cell secretome testing. This is going to combine transcriptomics with the single cell secretomics simultaneously. So from the same individual cell, you'll be able to get the transcriptome as well as the secreted cytokines from that individual cell. It should be very exciting. We expect that to start sometime next year as a project-based sort of beta test time, and then the following year should be available on your platforms. So that's the basics of the website. You can always go into the search function. So if you're looking for a particular item, you can search the website and try to find different data that way. So that's the website.
Thank you for showing us that.
My pleasure.
It's really great that you have the training sources there.
Yeah, they're actually really helpful. There's something that they made us go through when we're training to be field application scientists. So they really are super valuable. They even make the personnel that are helping you run this learn it. So
it's excellent. Thank you.
Yeah. So that's a great question. I don't usually get that question, which is why I wasn't ready for it, but I wish more people would ask it. And I think I'm going to start doing more of that in my presentations because it's a powerful resource. You know, there's a lot of information to be had from our website. So I really appreciate you asking that question. So we have a few minutes left. I'm going to stop sharing the screen here. So you guys can go all go to the website in all your spare time. Let me check in on the chat. No questions. Anyone else have any questions? Angel's asking some great questions. Okay.
So I have a question about your single cell transcriptome that have not come out yet.
Mhm.
So what does it, can you just give us a little bit more information about that? We have a 10x genomics box that we use for single cell transcriptomes and also Rhapsody.
And the sad reality, Angel, is that I really can't. It's a very new product, and so they're wisely, they don't tell the salespeople a whole lot so that we don't say things we shouldn't say yet, because they know if they tell it to me, it's going to fall right out of my mouth on the presentation. So I really, what I've told you is about what I know about it right now. Rose, I don't know if you have anything else that's legal to share, but
yeah, I'm in the same boat as you. Pretty much all I know is that we're going to do it, but I don't know any specifics about it, so I apologize. But when we do have specifics, we can definitely pass that on.
Right. The reality is, Angel, I wasn't even going to mention it in the presentation, except it was a giant picture right on the screen on the website, and I kind of felt like I had to say something. So,
all right, thank you.
You're welcome. Yeah, because it's a little early. Honestly, we're very excited as a company about having that data together. So the kind of data you're going to get from your 10x genomics, and I'm speculating here, but that kind of data combined with the data we're producing today on the functional phenotyping together seems to me like it would be very powerful. You know, kind of the way I think about it, what you're doing today with your 10x and what you'll be doing with flow cytometry and the bulk cytokine testing that you're doing, all good technologies that give us all good information. And they've sort of put us on a path for our research, right? I think we kind of, you're driving a car down a dark highway, and you can get that transcriptomic information like you get from the 10x, and it kind of lets you see the road signs, right? To see if I'm on the right path and I'm going in the right direction. But you don't have enough to understand, right? Just like in that glioblastoma study where they could understand the core set of mutations that were driving resistance, or I'm sorry, that should have been the drivers of resistance, and they were attacking them with therapy, but resistance continued. So what do we do next? Well, one of the things you can do is you can work with some bulk cytokine testing and flow cytometry. But what you're going to get is sort of a broad picture on the bulk and a very specific picture on the flow cytometry where, you know, you can fix permeabilize Golgi block and get some cytokines that are inside the cell, but you're not going to get the cytokines that are actually secreted. So you're still guessing a little bit. So in my little analogy, you're on the road, you've seen the road signs with your transcriptomics, and now with the flow cyto and the bulk cytokine analysis, you turned on your headlights and you can see a little bit farther down the road. They still can't see far enough, right? It's a dark night. So what I like to think of is what we bring to the table is you get that functional proteomics, where you get that functional phenotyping, where we're seeing for each individual cell what cytokines are producing and what effects those cytokines are having on the model you're studying. It's kind of like turning on the high beams on the car, so you're seeing even farther down that path, so we can see farther where to go. So these all work in a very complementary way. So I never want to throw the technology we're already using under the bus, because you can get some fantastic data from Cytop, and you can get some fantastic data from bulk analysis. Heck, we offer bulk cytokine testing on our platform. We do it, we think, a little better because it's automated and very low volume, but very useful tool still. But we're still not, you know, best as I can tell, we haven't cured cancer this week. So I think we still need to keep working at it, right? And so our technology complements what you already have to give you more information, to drive us farther down the road, to let us see the path ahead and get to that goal, right? So that we can get to those effective therapies that really can cure cancer and really will. I mean, you guys are doing the work today that's going to knock out cancer, that's going to take care of COVID, that's changing our world. This is just one more tool in your toolbox to do it, we think, a little bit faster and with unique data that we think is a little more insightful.
Thank you for that explanation. I appreciate it.
Oh, my pleasure. Hey, they pay me to talk, so I'll keep doing it. So we're down to 4 minutes left. I'll give you one last chance for one last round of questions before I let you all go to get on with your days. Okay. All right, as I am not seeing any questions in the chat, and I'm not hearing anybody piping up, then I will just say thank you very much. Rose and I sure appreciate you guys jumping on for the presentation. As Chris had mentioned, it's going to be recorded, so you can listen to me blather on over and over again if you like. And any questions that you have that you think of later, again, my email is in the presentation, and both Angel and Chris have all my contact information, so they can put you in touch with me as well. So I look forward to hopefully hearing from you all in the future. But thank you again, and have a great rest of your day, and we'll let you go.
Thank you all so much.
Thank you for coming for the great presentation.
It's our
Really
pleasure.
appreciate it.
Thank you for the opportunity.